Let's consider our second major class of membrane-associated protein as related to function. In this we will see a distinction between the protein transporters of our previous lesson and these membrane receptors. Membrane receptors are large multi-subunit proteins with molecular weights exceeding 100 kilodaltons. The primary distinction between receptors and transporters is that receptors exert their influence without actually moving a substance across the membrane. Instead, a substance on the outside of the cell binds to the external face of the receptor, which initiates an action within the cell. The binding of receptors and ligands is similar to that of enzymes and substrates in that they tend to be quite specific. The receptor binds its ligand in a specific site and in a specific way. Binding of the ligand to the receptor may also be interrupted by an inhibitor. Let's consider the receptor for LDL, or low-density lipoprotein particles. These are the principal carriers of cholesterol in the bloodstream. As the name suggests, these particles have both lipid and protein components. The protein component of these particles mediates binding to the LDL receptor on the surface of the cell. Binding of LDL to its receptor induces endocytosis, which releases the cholesterol component inside the cell. If the internal concentration of cholesterol increases, it inhibits synthesis of the LDL receptor, thereby preventing intake of more cholesterol. This leads to hypercholesterolemia, that is, an elevation of blood cholesterol levels. Another example of a membrane-bound receptor is that of human growth hormone, or HGH. As hormone binds, it causes dimerization of the receptor. The conformational change associated with receptor dimerization is transmitted to an intracellular domain which is bound to an enzyme known as Janus kinase 2 or JAK2. This initiates a kinase signaling cascade which serves to amplify and transmit the signal received by hormone binding. We will see the significance of the signaling cascades as we look more at metabolic pathways and control.